Okay, so we left off on capillary exchange. So remember, really thin walls. We're going to release oxygen, glucose, hormones, nutrients, all this stuff needs to go into the surrounding tissues. And then we're going to take up the waste, such as like carbon dioxide, ammonia from the surrounding tissues. So let's talk about a few of the mechanisms that are at work here to do that. And I guess it's good to highlight that water is going to move in and out depending on what the situation is. Okay, diffusion itself. So we talked about this a few chapters. Well, it's been a lot of chapters now, but substances are going to naturally move from an area of greater concentration to lesser concentration. So in this case, we're coming from the artery side of things, the arterial end. So we have a lot of oxygen-rich blood coming in. That oxygen-rich blood is traveling next to a tissue that has a very low concentration of oxygen. So we have a concentration gradient that is going to send oxygen from high concentration in the blood to the low concentration in the tissues. Vice versa, as we get to the capillary, as we're passing to the venule side of things, our tissues have a high amount of CO2 relative to the blood. So that CO2 is going to diffuse in and travel with the venous system. So remember, the big concept is greater to lesser concentration, and this is the by far the most important mechanism of capillary exchange. Um, so this just kind of were the words that we just talked through up here. Okay, filtration. So this is going to occur closer to the arterial end of our capillaries. So the pressure is higher inside the capillary compared to the surrounding tissue. So in this example, we see 30 millimeters of mercury. So that's a pressure number where we have 2 millimeters of mercury of pressure in the tissue fluid. So because we have more pressure here, it's going to force plasma, nutrients, all this stuff through the capillary wall into the tissues. So that is filtration. Okay, one last one here. We have colloid osmotic pressure, and then this is going to be on the venous side of our capillaries. So we have a relatively large drop down to 10 millimeters of mercury from the 30 on this end. So proteins in our blood can then exert colloid osmotic pressure because of this pressure drop. So albumin, that's the primary protein that you'll find in our bloodstream. It's going to draw fluid and waste products into the capillaries. And so about 85% of the fluid filtered at the arterial end. So this fluid that's being filtered at our arterial end, about 85% of it is going to come back on the venous end because of this colloid osmotic pressure. And I do recommend watching this animation on the mechanisms. It does help to see the visual of it, I think, and just to hear it one more time in a different way and watch kind of the movement of stuff while you do it. Okay, this is our artery map here. As you can see in the title, do not memorize this. This is very good to know in terms of your future endeavors if you're going into the healthcare field, but you do not need to memorize it for the purposes of this class. I do want you to know the primary arteries of the head and neck. So we have our external and internal carotid. So that is the branch off of our right and left common carotid arteries. And then we also have our vertebral arteries. They're going to arise from the left and right subclavian arteries, and they're going to extend up through the neck, cervical vertebrae. They actually enter the cranium. But our external and internal carotids, they are by far our largest blood flow supply, especially to the brain. So that's 80% of our cerebrum is the carotid arteries. Okay, so the circle of Willis, this is a mechanism that our kind of bodies have developed over time to ensure that the brain receives an adequate blood supply. So you don't need to memorize all the parts of things. But what you need to realize is that basically the circle is known as an anastomosis. That's a joining of arteries. And so they communicate with each other or meet with each other. And so the purpose of this is, say one side had an issue here. What if we had to cut off this side of the circle? The left side of the circle can still supply blood to the brain where it needs to go. So it's kind of like a backup system that if we have issues with one half, the other half can make up for that lack of blood flow. If we have issues with both halves or farther down the line here, then we're going to have issues. But in terms of the brain, the circle of Willis is a little bit of a safety system to make sure that our brain continues to get adequate blood supply. So that's the main thing you need to know about the circle of Willis, is that it's going to make sure our brain maintains its adequate blood supply. Okay, again, this is a map of a lot of our principal veins. You do not need to memorize this stuff. We'll highlight the hepatic veins in our jugular vein here shortly, but do not memorize every single vein. It's just nice to be able to see it. And like I said, in the future, if you plan on continuing with anatomy stuff into the medical field and all that, then it definitely is something that is good to start working on. All right, so just like the arteries of the head and neck, I would like you to understand the principal veins of the head and neck. So the internal jugular vein is what gets most of its blood from the brain and the face. And so that's going to go down and eventually merge with the superior vena cava. The external jugular vein, that rather than getting it from the brain and face, it's going to be the scalp, facial muscles, more superficial stuff. But those two, internal and external jugular, are going to be primarily what drains down into the superior vena cava. The vertebral vein is going to drain our cervical vertebrae, some of our spinal cord, some of our neck muscles, but the internal, external jugular are definitely the bigger capacitance vessels of the head and neck. Okay, back to the pulmonary circulation. So this is our routes of blood to and from the lungs. So remember, we're going and you see these numbers of steps here. That is on page 324 in terms of a written description for every single step. We're going to talk through it a bit. It's also under the notes section of this slide in the PowerPoint if you want to actually read through it as well. This animation talks you through all 5 steps. But I'm going to kind of give you the big picture of what you need to know. So we have, remember, right atrium is receiving our blood back to the heart. We're going into the right ventricle, and then that right ventricle is going to pump through the pulmonary arteries. The pulmonary arteries are going to carry blood to both the right and the left lung. So the body's putting CO2 in, we're getting oxygen out. So we have the deoxygenated blood passing into the lungs. It's going through the capillary network here, and then we're going to get the oxygen supply, and then that's going to go back into the left side of the heart. Obviously, the left atrium receives that. It's going to go to the left ventricle, and then we're going to get that to pump out to the body. So let's go ahead and do this. Let's drop this out. Okay, let's talk through this together just so everyone is definitely on the same page. Okay, so as I said, our blood's going to leave through the right ventricle, travel through the pulmonary trunk. That's going to be the pulmonary arteries, right and left. You don't need to necessarily memorize this lobar arteries part of things, but our lungs have different lobes. We'll talk about that in the actual respiratory chapter. But each lobe of our lung is going to have an artery that brings blood to that specific lobe. We're going to then branch into our capillary beds. Our capillaries surround the alveoli. So those are our air exchange sacs. We'll talk more about those. They're very important, but we'll go into detail on that in the respiratory chapter. And that's where our oxygen and carbon dioxide exchange occurs. And then our capillaries form our venules. Those venules are going to merge to form the pulmonary veins, and those veins are going to go back into the left atrium. So if you think about it, our capillary network almost looks flipped here compared to what we were looking at in the earlier slides, because the arteries and arterioles are carrying deoxygenated blood while the veins are carrying oxygenated blood back to that left part. Okay, let me get these out of my way here. All right. So the systemic circulation, as we said, the big goal is we need to get oxygen, nutrients, and everything that our cells and tissues need to them, and remove the waste from their metabolic processes. And so all of our systemic arteries are going to arise, whether it's directly or indirectly, from the aorta. So our aorta actually has a few different parts. So we have the ascending aorta. That's that first part. Where it loops here, that's your aortic arch. And then you have your descending aorta, which travels all the way down through here. Okay, the hepatic portal circulation. So hepato is the term for the liver. So hepatic portal circulation, we're talking about our liver. So veins of other abdominal organs, they go into the inferior vena cava. But certain digestive organs and our spleen actually don't empty into the inferior vena cava directly. They're going to send their blood through the hepatic portal vein, which, like I said, that travels to the liver. So our liver is really, really important in terms of a filtration and cleaning system for our blood. So it's going to modify the blood before sending it all the way back to the heart. So it's going to remove things such as excess glucose, alcohol, byproducts of medications. So when you hear about cirrhosis of the liver from alcoholics, it's because the liver is working so hard all the time to try to remove the excess alcohol from the bloodstream that it starts kind of dying off. In terms of medications, so say we're allowed to take an ibuprofen every 4 hours or so. That's because the half-life of the medication, or how quickly our body is cleaning that medication out, is about 4 hours. So that's being done by the liver. All right, here's a little bit of an illustration of this. So don't worry too much about memorizing every single piece here. I need you to get the big picture that we're sending blood to the liver to be cleaned, basically, before it goes back to the heart. So the stomach, the spleen, both the large intestine and small intestine are all part of this hepatic portal circulation. So again, there's an animation that walks you through this a little bit as well. So basically, the blood from the capillaries of these different organs are going to flow into our superior mesenteric vein and inferior mesenteric vein. The portal vein is actually going to channel the blood into the liver itself. And so this is an area that we have a lot of sinusoid capillary networks, because we need that high exchange rate, and bigger substances are passing through. So blood's going to then flow out of those sinusoidal capillaries in the liver, and that is then going to return to the inferior vena cava to the heart. So it's still getting to the inferior vena cava. It just has to pass through the liver's cleaning system first. All right, we're going to talk just some of the principles of circulation, or kind of why blood flows the way it does. So the primary principle of blood flow, or the key for blood flow, is that our circulatory system creates this pressure gradient. So you can see on the map here, on the y-axis, if you want to call it that, you have your blood pressure itself. And then here we're going through kind of as we get away from the heart and eventually veins wise back towards the heart. So your systolic blood pressure, that's the pressure when the left ventricle contracts. That contraction is known as systole. So that's your systolic blood pressure, is the pressure when your left ventricle contracts. So normal is considered 110 in terms of millimeters of mercury. They used to say 120, but us Americans have issues with high blood pressure, so they've been a little more strict. So they've dropped that norm down to 110. So our diastolic blood pressure, that's the pressure when the ventricle relaxes, which is known as diastole. And so that, I'm sorry, I didn't put that underneath there, but the normal for that one is about 70 millimeters of mercury. So 110 over 70 would be considered ideal blood pressure. And in general, the further away you move from the heart, the more our blood pressure is going to decline. And that's what is going to create that pressure gradient. So we're going from high pressure to low pressure. Okay, your pulse, your heartbeat or pulse is what you feel when an artery comes close to the skin surface. So I'm sure throughout school you certainly learned you have your radial artery in your wrist is a very easy one to feel. If you go kind of just distal to your wrist, or I guess just proximal to your wrist on your thumb side and kind of find a little soft spot, you should feel a pretty good beat there. You also have your carotid pulse, which is quite easy to feel. If you go and kind of find the to lateral to your neck, you can find a bit of a soft spot and you should get a pretty good beat there. So each pulse corresponds to your heartbeat. That's why we can track heart rate peripherally. We don't necessarily have to listen to the heart to get heart rate. And so we can make judgments based on whether this is fast, weak, if it's irregular, meaning that, say, instead of bump, 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 you're getting bump, 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 bump. So it should be kind of a normal rhythm as well. And so you guys do need to understand this chart. It's relatively simple, but you should understand how different factors are going to affect our blood pressure. Last chapter, we talked about cardiac output. So increased cardiac output is going to result in increased blood pressure. Decreased cardiac output is going to result in decreased blood pressure. So that goes hand in hand with our next one. So decreased blood volume is going to result in decreased blood pressure, whereas increased blood volume is going to result in increased blood pressure. So in general, if we're introducing more of a pump or more blood into the system, our blood pressure is going to go up. So resistance is another really important part of things. So an increase in resistance is going to decrease blood flow, but increase blood pressure. Decreased resistance is going to increase blood flow, but decrease blood pressure. So the more resistance the blood has, the more difficult it is to flow. And so it's going to have higher pressure. If we decrease that resistance, the blood's going to flow much easier, but we're going to have less pressure overall in the system. Okay, so that's what we were just talking about here, this peripheral resistance concept. So resistance to the flow is going to result from the friction of blood against the walls of the vessels. And so the amount of friction is going to depend on a few different factors. So we have blood viscosity, or how thick our blood actually is. So it makes sense that if our blood is thicker, we're going to have increased resistance. Vice versa, if we have thinner blood, we're going to have decreased resistance. Vessel diameter wise, we have vasoconstriction, or when our vessels get smaller, that's going to increase pressure and decrease flow. Vasodilation, where the blood vessel is expanding, we have an increased diameter of the vessel. That's going to decrease pressure, which will allow for an increase in flow. Okay, blood velocity, or how fast our blood is flowing, and that's going to be primarily affected by vessel diameter. So if you think the biggest diameter artery we have is the aorta, so the fastest blood flow is in the aorta. So it slows a lot in the capillaries for a couple of reasons. One, we're farther away from the left ventricle, so we have less pressure pushing the speed. Friction, kind of that peripheral resistance that the blood has had to travel past, is going to slow things down. We have smaller diameter. We just said higher, kind of greater diameter, faster blood flow. So we have smaller diameter. We also have a lot more vessels. You see, as we travel through, we're kind of spreading out, and you'll see the velocity is going to drop down a lot. And then as we start combining our venules into veins, we start getting a slight increase again. So the blood has to spread out, and that is really good for the actual exchange side of things. We don't want it flying through our capillaries because we need the time to exchange our nutrients, gases, waste, all that stuff. Okay, so vasoconstriction again, just to illustrate it a bit. This is what actually adjusts the diameter of the vessels. So this is our tunica media layer that is at work here in terms of actually causing the constriction dilation. Remember back, the tunica intima is what is going to release our chemicals, but the tunica media is what has the muscle to constrict and relax. So increased pressure and decreased flow with vasoconstriction. Vasodilation, we're going to have decreased pressure, but increased flow. So vessels get smaller, resistance goes up, pressure goes up, flow goes down. Vessels get bigger, pressure goes down, flow goes up, resistance goes down. Okay, talking about the regulation of our blood pressure, again, not something that we make kind of a conscious effort. Oh, my blood pressure's high, let's lower it. This is something that the body does, again, with the autonomic nervous system. So decreased blood flow is going to produce hypoxia, meaning we don't have enough oxygen. Hypoxia produces waste products, and then the accumulation of that waste is going to stimulate vasodilation. So remember, vasodilation, vessel gets bigger, we get more blood flow, more oxygen, we can remove those wastes. Endothelial cells and platelets, they're going to secrete chemicals. That's that tunica intima layer that can change our vessel diameter. So when the tunica intima releases nitric oxide, that's going to be the trigger for vasodilation. And then there's another chemical substance known as endothelins that is a vasoconstrictor. So that's how that's that chemical signal from the tunica intima that tells the tunica media what it should be doing. We also have a neural regulation of blood pressure. So baroreceptors, which are pressure receptors, they're located in our carotid sinus and aortic arch. They can actually detect what our blood pressure is and if it changes from our normal set point. So this is still, again, kind of like a homeostatic or a maintenance of homeostasis mechanism. So they're going to relay information to the vasomotor center. That's in our medulla, up in the brainstem, and that's going to signal our autonomic nervous system to respond accordingly. So increase in blood pressure, we're going to get parasympathetic impulses. Remember, that's kind of our calming system. We'll get vasodilation, decrease in blood pressure. Decrease in blood pressure, we get sympathetic impulses. That is our kind of stimulant system. It's going to cause the muscles to constrict. That's going to increase our blood pressure. So if you think way back, this is a negative feedback loop. So increased blood pressure causes the body to try to decrease it. Decreased blood pressure causes the body to want to increase. So we're trying to maintain a set point. You can also have hormonal regulation of blood pressure. So there's a number of hormones that actually raise blood pressure, and you don't have to necessarily understand exactly how this happens a ton, but you do need to know which ones are going to vasoconstrict and which ones are going to vasodilate. So that is definitely important. So the ones that will raise blood pressure, which is a majority of these, you have renin, angiotensin 1, angiotensin 2. And so they are vasoconstrictors, and they also cause water retention. So the angiotensin system is a huge control of blood pressure. You have aldosterone, that's going to stimulate the kidneys to retain sodium, and kind of by default, that's going to result in more water being retained as well, again, raising blood pressure. ADH, antidiuretic hormone, that's going to promote vasoconstriction and water retention. And then we have epinephrine and norepinephrine, that's also going to cause vasoconstriction. So some of them are vasoconstrictors and result in water retention. Some are just vasoconstrictors, but all of those in that top category are going to raise blood pressure. Atrial natriuretic peptide is going to cause blood pressure to fall. So that causes vasodilation and the kidneys to retain. Oh, I'm sorry. I don't think I think I messed that up for you guys. The kidneys are actually going to release sodium. So that's going to cause blood pressure to fall. Okay, last couple slides here, guys. Our skeletal muscle pump. So this is our skeletal muscles help our venous system work a little bit more efficiently. So when we get contraction of our leg muscles, they're going to squeeze our veins, and that helps push the blood higher up. And then we talked earlier about how veins have these one-way valves. They're quite tiny in this picture. But if you see, the calf muscle here is squeezing blood up the vein, and this valve here is closed so that when the muscle stops contracting, it doesn't open back up. So same thing here. You see squeezing, open, pushing up. You see here when it relaxes, these are going to close so that that blood can't flow farther down. So that's part of the way we help our venous system pump blood back against gravity. Our respiratory pump also helps. So this is more of a pressure change. So breathing is going to promote venous flow in the thoracic and abdominal cavities due to changes in pressure. So as an example, when we breathe in, we get a drop in the diaphragm that results in a drop in our chest pressure. And so that creates a pressure gradient that promotes blood flow in the thoracic and abdominal cavities. And that is, I believe, our last one. Let's just talk a little bit more about the respiratory pump for just a second. Let me pull this part up for you guys. So just to make this clear. So during inhalation, our chest is going to expand. The diaphragm is going to move down. Said we're going to have a drop in our chest cavity pressure, but a rise in our abdominal pressure. So that rising pressure down here from us breathing in is going to push blood up into the inferior vena cava and towards the chest cavity. And then that lower pressure in our chest cavity or thorax is going to actually bring the blood to the heart. So our skeletal muscle pump and our respiratory pump are two systems our body uses to help aid venous return. All right, that is the last one for this slide or for this lecture. So I will be uploading this shortly for you guys. Thank you for listening and have a good day.